Hi everyone. In this video, we will discuss about the principal mechanism of the adaptive immunity against the viruses. When viruses attack our body, they will easily infect our body cell and making all these body cell to start to replicate the viral DNA and also producing all those viral protein. So in a very fast pace, the virus can make our body cell to produce lots more virus. And all of this new virus that is being replicated can easily infect more cell and releasing more virus at the same time. Our innate immunity is not acting fast enough to eliminate all of this virus. The natural killer cell will be outnumbered. They cannot eliminate all of these virus infected cell, so causing there are lots more virus infected cell and also the virus in our body. That's why we will depend heavily on the adaptive immunity to eliminate all these virus and also the virus infected cell. The first principal mechanism in the adaptive immunity, it would be the antibodies. Antibody will work very well to eliminate all these virus that is still at the extracellular stage. So meaning that the antibody will mainly eliminate all those virus that occurs outside the cell or haven't infect our body cell. So how these antibody are being produced, how the humoral immune response are being activated. The process of the antibody production can be divided into three stages: first stage, second stage, and the third stage. The first stage occurs in two parts. The first part would be on the APC and the T cell, and the second part would be on the B cell. But please remember, both of these parts in the stage 1 occurs at the same time. So in stage 1, when the virus-infected cell is being attacked by the natural killer cell of the innate immunity, so some of these virus-infected cells will undergo apoptosis induced by the natural killer cell. So this cell will be dead, and there will be all the remnant, cell debris, and the viral antigen. So all of these debris and remnant will be engulfed by the phagocyte here. So the phagocyte normally can be the macrophage or the dendritic cell. So the macrophage will engulf them, digest all of these, they will digest it, and then digest and extract this antigen. So after they extract the antigen of this virus, they will present the viral antigen on the surface through the MHC class 2 molecule. So later on, these phagocytes will act as the antigen presenting cell to present their antigen on the MHC class 2 to the naive CD4 T cell. So the naive CD4 T cell will use their T cell receptor here to recognize the viral antigen that is being presented on the MHC class 2 molecule of the APC. So after the T cell receptor recognize the antigen, then next, the naive CD4 T cell will have the CD4 marker. So this CD4 marker will bind to the MHC class 2 molecule to confirm that. And after this confirmation, then next, they will need one more round of the co-stimulation. The co-stimulation occurs in between the APC and the naive CD4 T cell will fully activate this naive CD4 T cell to undergo the chronoselection. Normally, the co-stimulation molecule or the so-called co-stimulator, will be the B7 molecule on the APC and the CD28 receptor on the T cell. So after the recognition of the antigen plus the co-stimulation, the naive CD4 T cell will be fully activated and undergo the chronoselection to produce the effector cell. The effector cell is the helper T cell. Of course, the chronoselection will also produce the memory helper T cell. But this memory helper T cell only work for the immunological memory, which means when the body is exposed to the same virus for the second time. As for now, it is the effector cell that will work on. The helper T cell will be fully activated, and now it will enter the stage number 2. But before that, you have to look at the second part of the stage 1, which is the B cell. So at the same time in the stage 1, when the phagocyte engulf this viral antigen and the cell remnant, the B cell will also expose to the same viral antigen. So this same viral antigen will be binding to the B cell receptor on the surface of this naive B cell. When the naive B cell recognizes the antigen, then this B cell will be activated. The B cell activated, then they will internalize this antigen, this viral antigen, they will internalize it and later on present it on the MHC class 2 of this B cell. So now this activated B cell will present the viral antigen on their surface of the cell through the MHC class 2 molecule here. And now this activated B cell can act as the APC. They will act as the antigen presenting cell to present this viral antigen to the helper T cell just now. So in stage 2, the activated B cell that can act as the APC now, and also the helper T cell, the activated helper T cell, will come to find each other. They will come close to each other, and now the B cell will present, they will act as the APC to present the viral antigen that's presented on their class 2 MHC molecule to the helper T cell. And again, the helper T cell will use their T cell receptor here, the T cell receptor, to recognize the viral antigen again. So after they recognize the viral antigen, then next, the CD4 marker of the helper T cell will bind to the class 2 MHC molecule here to double confirm. So after the binding of the CD4 marker to the MHC class 2 molecule and also the recognition of the antigen, then this helper T cell will release the cytokine. This cytokine will bind to the B cell and cause the B cell to be fully activated. This fully activated B cell will undergo chronoselection in order to produce their effector cell, which is the plasma cell, and also some memory B cell. But the plasma cell is the one that is going to immediately produce the antibody against the virus. So in stage 3, the plasma cell, once it's being produced, they will start to release all of this antibody, and the antibody will go and bind to this extracellular stage of the virus. When the antibody binds to the virus, then this virus will be neutralized. This virus can no longer attack the new body cell. Some of the antibody is capable of causing the agglutination of the virus as well. And by doing this, the antibody will eliminate this extracellular stage of the virus. Now, please remember here, antibody is only or mainly acting on those virus that is outside the cell. They are not effective at eliminating all those virus-infected cell that is still able to produce the new virus. So in order to eliminate these virus-infected cell, we will need the second mechanism of this adaptive immunity, which is the cytotoxic T cell. The activation of the cytotoxic T cell will start with the APC as well. It will start with the phagocyte engulfing the remnant and then activation of the helper T cell as well. So the whole process will involve 4 stages: 1, 2, 3, and 4. So let's look at the stage number 1. So when the virus-infected cell is being killed by the natural killer cell, so there will be some remnant and also the viral antigen. The phagocyte is going to engulf this remnant, and then they will digest it. The phagocyte will later digest of this remnant and also the viral antigen, and then they will extract the viral antigen and present them onto the class 2 MHC molecule. At the same time, these APC will also able to present their viral antigen on their class 1 MHC. Normally, these phagocytes would be the dendritic cell, because the dendritic cell will acting as the antigen presenting cell to activate the helper T cell and also the naive CD8 T cell at the same time. So these dendritic cell, after engulfing the remnant, they will act as the antigen presenting cell and present the viral antigen on both the MHC class 1 molecule and also the MHC class 2 molecule at the same time. So when the dendritic cell acts as the APC here, the helper T cell will come and recognize the viral antigen that's being presented on the MHC class 2 molecule, the MHC class 2. So these helper T cells are the effector cell. They will directly use their T cell receptor to recognize the antigen that's presented on the MHC class 2 molecule. And at the same time, their CD4 marker of the helper T cell will bind with the MHC class 2 molecule. So with this confirmation, then the helper T cell is ready. And at the same time, there will be viral antigen presented on the class 1 MHC. So this viral antigen on the MHC class 1 will be recognized by the naive CD8 plus T cell. So this naive CD8 T cell is actually the precursor cell for the cytotoxic T cell. This naive CD8 T cell will use their T cell receptor to recognize the viral antigen that's being presented on the MHC class 1. After the recognition of the antigen through the T cell receptor, then next, the naive CD8 T cell will use their CD8 marker to bind with the MHC class 1 molecule. And with this double confirmation, then next, the co-stimulation should occur. within the naive CD8 T cell and the APC. And again, normally this co-stimulation will involve the co-stimulator like the B7 on the APC and the CD28 on the naive CD8 T cell. So with the recognition of the antigen and also the co-stimulation, these naive CD8 T cell can be now activated. But to be fully activated and undergo chronic selection, we will need to go into the stage 3. So after the activation of the naive CD8 T cell through the antigen presentation on the MHC class 1 molecule by the APC, and also the activation of the helper T cell through the antigen presentation on the MHC class 2 molecule on the same APC. Now, this activated helper T cell will release the cytokine, and this cytokine will be accepted by the naive CD8 T cell. The cytokine will fully activate the naive CD8 T cell to undergo chronic selection. So after the chronic selection, the naive CD8 T cell will produce the effector cell, which is the cytotoxic T cell, and also the memory cell. The cytotoxic T cell is the effector cell that will immediately act on the virus infected cell. These cytotoxic T cells are activated. And once they are activated, they no longer need any co-stimulation to start the killing. Because this cytotoxic T cell, which is the effector cell or which is activated, will have the killing mechanism that can kill any virus infected cell that they recognize. These cytotoxic T cells will now go into the stage 4, which is they start to search around. They start the searching and start to find out all those virus infected cells with the same antigen. So when the cytotoxic T cell recognize the antigen that's being presented on the MHC class 1 of this infected cell, then they will go and bind and start the killing mechanism. All the virus infected cell, after being infected by the virus, they will start to present the viral antigen on their surface through the MHC class 1 molecule. So this antigen that's being presented on the MHC class 1 molecule will be recognized by the T cell receptor of the cytotoxic T cell. After the T cell receptor recognizes the antigen, then next the CD8 marker will bind to the MHC class 1 molecule on the surface of this infected cell. And once there is a confirmation, then this cytotoxic T cell will release the perforin and also the granzyme. The perforin is going to create the pores on the membrane of this infected cell, while the granzyme will enter the cell and induce the apoptosis of this infected cell. This process will be more specific and faster and more efficient as you compare to those of the natural killer cell. So that's why the cytotoxic T cell is able to eliminate all of these virus infected cells. And when the virus infected cells are killed, then again, the phagocyte will come and clean up all of these remnants. So with that, these are the two principal mechanisms of the adaptive immunity against the virus. And compared to the innate immunity, the adaptive immunity is the key or play a key role or is very essential that will eliminate this virus infection.